Thank you again, and it's a real pleasure to have been invited by Miltenyi Biotec to give a presentation about our work on tumor microenvironment. And in particular today, I'm excited to share with you our work on perivascular macrophages and their roles as kind of gatekeepers of the immune landscape of the tumor microenvironment. So I have some disclosures. So obviously, we're all aware that obviously the field of the tumor microenvironment has really kind of blossomed, and with all the new technologies such as single cell RNA sequencing, we're getting a really deep understanding of the heterogeneity of the various populations of stromal cells, be them immune cells or mesenchymal cells within the tumor microenvironment, which collectively contribute to this kind of familiar of different cell types, cytokines, extracellular matrix, which encompass the microenvironment of a cancer, which is supporting tumor progression. Now, my lab has taken indeed a focused look on tumor-associated macrophages. We really found these cells interesting. Because probably one of the most plastic cells of the immune system means that they are very amenable and they can be exploited by cancer to facilitate various facets of cancer progression, such as encouraging angiogenesis with the kind of new blood vessel growth into cancers. It can facilitate the metastatic process associated with kind of the invasion and migration of tumor cells, the epithelial to mesenchymal transition of these cells, also making these cells more motile. And an area, obviously, with my background as an immunologist, I've taken a particular interest in is this kind of concepts of their role in immune suppression, their ability to shut down kind of anti-tumor immune responses, which could potentially eradicate the disease. Now, many on this call will be familiar with, obviously, macrophages in cancer. Yes, they are a prevalent cell type which gets absorbed into cancer. But obviously, their heterogeneity is something that we really only started to get a deep dive on as our abilities to look at these cells using kind of non-subjective approaches like RNA sequencing, single cell RNA sequencing. But many will be familiar with this kind of M1 and M2 paradigm. So the majority of macrophages that are found in tumors are recruited as monocytes from the bloodstream. And these cells, once within a tumor microenvironment, differentiate into macrophages, but also polarize. And this is a key concept. This idea of polarization, where a cell is starting to equip itself with specific effector function based on the environment that cell finds itself within. And macrophages are particularly good at this. So let's say, in the context of just inflammation, a monocyte differentiates into a macrophage in a site that has very clear pro-inflammatory cues, such as high interferon gamma, bacterial ligands, such as lipopolysaccharide, and this leads to a very pro-inflammatory phenotype of our cells. They secrete pro-inflammatory cytokines. They make receptors and molecules associated with a kind of a pro-inflammatory immune response. However, it's not always wanted that you want a purely pro-inflammatory kind of context. And actually, sometimes you want these cells to display a kind of a repair process, for maybe potentially modulating immune responses in the context of, for example, wound healing, to allow a wound to heal quicker. These kind of environments where we don't see pathogenic signals, it's sterile inflammation, such as what we see in cancer, but where you might find signatures of maybe Th2 responses, such as interleukin-4, IL-13. Now, this can evoke a relatively anti-inflammatory program within our macrophages. This can involve the secretion of immune suppressive cytokines. It can lead to the expression of enzymes which can modulate the immune response. So ultimately, this is a paradigm which is very familiar to a lot of people. Now, when I first established the tumor immunology group back in 2012, one of our first experiments was to set up a microRNA screen. We really wanted to get deep into the understanding of how similar then is this M1, M2 cell-like to a tumor-associated macrophage. And this is a slightly archaic technology in the fact that this is a microarray data, but I love this data set because it's a data set that we've always kept coming back to. It's actually provided us a lot of meaningful information about these cells, which has, I say, served the test of time. So if we look at this, basically every dot in the same color represents a biological replicate. And we used bone marrow-derived macrophages to derive an M0 macrophage, which is just a CSF1 basal macrophage. We've not provided any kind of polarization cue per se. If we stimulate to an M2 phenotype using IL-4, we have those cells. The M1, we used interferon gamma and interferon gamma and lipopolysaccharide. Now, as a comparator, we did a bulk sort on tumor-associated macrophages from two different models of breast cancer. These are subcutaneous models. The red dots represent EO771 tumors, and the blue represents TAMs that we've taken out of 41 tumors. Now, you can see on this PCA plot that these actually don't really particularly overlap with either M1 or M2 or the basal macrophages, but actually they sit quite close in clustering to one another. Now, this for me was an exemplification of this idea of actually there are some forthcomings in this model in talking broadly about M1 and M2 macrophages, but obviously to some degree there is overlap. So obviously even the data back then was alluding to this idea of M1-like, M2-like cells. But if you look at the red ball on the right-hand side, these are the differentially expressed genes. And the red at the bottom are the key genes which are uniquely associated to tumor-associated macrophages. And this was over 1,667 genes. So, very much this idea that there's a lot of unique features which aren't captured in the M1, M2 paradigm. Now, today's I'm going to be talking a lot about our work in the spontaneous murine model of breast cancer. These is where all of the data that I will present to you hereafter come from. So it's a spontaneous model of breast cancer. This is coming from a mouse whereby the mammary-specific promoter, the MMTV promoter, is driving a viral oncogene, PYMT, which is driving transformation of the mammary epithelium. So after about the 70 days of age, our mice start to develop lesions whereby we can track tumor growth thereafter. We like this model because you think it's a relatively good trade-off between a close clinical parallel with tumor latencies that we can work with for our experiments. The healthy mammary epithelium in these mice goes through all of the stages that human cancer development does, through kind of hyperplasia, through to kind of bona fide tumors, which actually also can be metastatic. So with the dawn of single-cell RNA sequencing and my labs kind of focus in these areas, we really couldn't resist the idea of actually running a single-cell RNA sequencing analysis of a bulk, broad, sorted population of tumor-associated macrophages from our PYMT model of breast cancer. So we were gating cells, as I say, very broadly, trying to capture all variables of the TAM kind of subset. So this is a broad F480 high gate, which is a marker in mice associated with macrophages, and CD11B, obviously the broad myeloid marker that is associated with all myeloid populations. So these sorted cells, when we conducted a kind of clustering, we were able to identify 8 distinct phenotypes of tumor-associated macrophages in these mice. Now, we started getting, obviously, we didn't really know where to start on this. That was a lot of information, a lot more than we'd actually thought we'd be able to kind of work through. So we started developing this idea, actually, how similar are these? And obviously, there's some really neat programs for analyzing these data. And then we got interested in trajectory analysis, considering that these clusters are likely tightly associated potentially with other relatives. We wanted to get an idea if we could map out predicted TAM development within the tumor microenvironment. So we conducted a slingshot analysis whereby we were able to define there was 3 directions through which these TAMs seem to be polarizing. And we can map them out on this schematic here on the right-hand side, where TAM1 represents the most naive TAM, likely the least polarized, going to a point where we have terminal differentiation states, TAM7, 6, and 4. To understand what these terminal states are, these became the cells we really wanted to focus in on because they likely represented the end product, the TAMs that were probably playing the greatest role in the progression of the kind of the disease. And actually, this is where we took our focused look. Now, interestingly, we found that TAM7 has an M2-like signature, TAM4, which is the yellow arm at the very on the light's left side, actually had an M1-like signature. Now, I won't go in that today, but I find it interesting that obviously you can have both anti-inflammatory and pro-inflammatory macrophages in one given microenvironment. But the cell that really attracted us was this TAM06. It didn't really fit into the M1, M2 paradigm. It seemed to be something else. So we looked at the kind of pathway analysis. What was actually going on within this cell? What could we learn about it? And what we found is that there was this kind of overarching kind of emphasis on pro-angiogenic pathways. It was associated with modulation of inflammation. And that's why we thought, well, it didn't seem very well characterized. And we wanted to see if we could find this at the protein level. I mean, we could start identifying where it sat spatially in the tumors. So we used the single-cell RNA-seq data to understand what markers could guide our ability to identify these cells. They expressed the highest levels of MRC1, which is also CD206, macrophage mannose receptor. They also were selectively expressing the surface receptor LIVE1, which is a hyaluronin receptor. So we explored at the protein level using enzyme-digested tumors from the PYMT mice, where we stained for these protein markers to see if we could identify this population. And we found that we could quite clearly identify it as an MHC class 2 low, CD206 high cell. They sit as a very discrete cluster as TAM06 that I've marked out here in the middle. And if we look at the LIVE1 expression on the cell surface, it was very evident that indeed this is our population of cells that we wanted to find. The single-cell RNA-seq gene predicted expression did map to the protein level. They were CD206 high, LIVE1 positive. So we turned to confocal microscopy to map out where in the tumor microenvironment these cells resided. We used F480 again as a broad tumor-associated macrophage marker, the subset-specific marker LIVE1, and the white events that I'm showing you here on the far left represent co-localization of pixels between F480 and LIVE1. So this is our LIVE1 positive TAM subset. These subset of TAMs actually sat proximal to blood vasculature, which is what is being mapped out here in green. So it was very clear that we were working with a subset of cells which is associated with blood vasculature, making them a perivascular macrophage subset. So we wanted to question, well, where did these cells come from? What was the signal that was actually deriving them? You know, we're all familiar with this idea of polarization, but what was the signal causing this subset of TAMs to develop? We used a program called Ingenuity, which is a QIAGEN package, which took the transcriptional kind of differentially expressed genes of this TAM population, and it predicted what an upstream mediator could be for explaining that gene signature. It gave us a very strong steer towards interleukin-6. So we tested in our murine model where indeed it may be interleukin-6 was a major polarizing force behind these cells. So we crossed the PYMT mouse onto an interleukin-6 knockout background, and we analyzed the tumors. Now, whereas we could find the LIVE1 positive TAMs in the wild type mice, in the absence of IL-6, there was a clear loss of LIVE1 positive TAMs, and we quantified this across multiple tumors. As such, what we were finding then that indeed this TAM subset was developing in response to interleukin-6. So where was that IL-6 signal coming from? So for this, we turned to an RNA-scope experiment. We looked for mRNA using RNA-scope for interleukin-6, and we knew the cells that were in the perivascular niche, and we stained up for a variety of cells. But what I'm presenting to you here was, one again, a very clear-cut outcome. The IL-6 was sitting very tightly with endothelium, which is CD31, which are the green cells you're seeing in the top left. There was a very close concordance, and obviously that concordance is mapped onto the yellow that you're seeing in the bottom right. And as you can see, this endothelium that's expressing IL-6 sits very close to these LIVE1 positive macrophages. So this is the likely source of the polarization signal. Now, anecdotally, when my lab were studying these cells, and I always love it when observations just come out of kind of unpredicted kind of areas, and it's just purely researcher observation of what they were seeing down the microscope. And what was evident, it wasn't as we would have predicted, actually, that, you know, these TAM populations probably would sit across all of the vasculature within the cancer. But actually what was coming out as this anecdotal observation is that these live-1 positive TAMs were actually seen to be accumulating into nest structures, multicellular nests, which got us really interested in the idea that these cells and subsets maybe potentially had a communication axis between them that was causing them to form these structures. And we're all aware that nature seldom does anything by accident, so this structure could have actually been biologically important for the function of these cells. So as I said, I mean, we knew that these macrophages were polarized by interleukin-6. So rather than try and dissect a large amount of information, we took a very focused look on the hypothesis that the chemokine receptor that was likely responsible for these nests was probably also regulated by interleukin-6. Now, we had a very nice data set whereby we had a comparison between a basal macrophage and then an interleukin-6 derived macrophage, and we could screen for the chemokine receptors. And probably of all the microarray transcriptomic data that we've ever analyzed, this is probably the most clear-cut observation that we've ever had out. And the only chemokine receptor that was regulatable by IL-6 was CCR5. Now, this worked quite nicely because it was logical. We looked at the cells within the perivascular niche, and really the live-1 positive TAMs were the only expressor of CCR5. They also expressed the ligands CCL3 and CCL4, which meant that these cells had both the chemokine receptor and were making the ligand, which provided a potential communication axis to allow these cells to find each other in the perivascular niche. So we questioned whether CCR5 could potentially be playing a role in the formation of these nest structures. So we crossed our PYMT mouse onto a CCR5 knockout background, and we looked at the structures of these nests. In the absence of CCR5, what we were finding was that indeed these nests were starting to dissolve. We had the same numbers of live-1 positive TAMs, but they were drifting away from the blood vasculature. They were also drifting away from each other. So this CCR5 communication axis was actually playing a major role in the structure of these nests. Now, we're particularly interested also by these cells is because they selectively expressed another enzyme called hemoxygenase 1. And it's an enzyme that my lab has been particularly interested in for a while. Now, we wanted to kind of take a bit of a look in to see if we could find a biological mechanism associated with these cells, maybe potentially associated with these nest structures. Now, my lab have a hemoxygenase 1 reporter mouse, which reports GFP expression when hemoxygenase 1 is being expressed. And using this mouse crossed onto a PYMT background, we could also demonstrate that the live-1 positive TAM subset was the sole expressor of hemoxygenase 1. In fact, it was the sole expressor of all of the tumoral cells. So it's the major source of this enzyme. And we wanted to probe this idea because my lab had been working on this enzyme and its effector function in the tumor microenvironment, that these effects could be attributed to our perivascular macrophage. So hemoxygenase 1, for those who aren't familiar, is an enzyme which degrades heme to generate the catabolites carbon monoxide, biliverdin, and ferrous iron. And carbon monoxide from an immune perspective is particularly interesting because it is associated with immune suppression, suggesting that expression of this enzyme could play a role in suppressing anti-tumor immune responses. So we conducted an inhibitor experiment. There was a very good inhibitor for hemoxygenase activity, and we wanted to inhibit this enzyme within live-1 positive TAMs. Inhibiting this enzyme, in the absence of any other additions of therapy, had no effect on tumor growth. So we were starting to kind of question a little bit deeper. I mean, maybe we need to evoke an immune response to start seeing these effects. Now, if we analyze these tumors, it's actually very apparent that the PYMT model is immunologically cold, very similar to what has been seen clinically, where some patients don't have a prevalent influx of CD8 T cells to the tumor microenvironment. So we were actually thinking on this idea, maybe we needed to evoke an immune response before we could start understanding whether these perivascular macrophages might be having any immune modulatory role in the tumor microenvironment. So for this, we turn to chemotherapies, because obviously this role of chemotherapy in shutting down kind of cell cycle or sort of machinery of the malignant cells is one arm, but actually there's this beautiful growing literature that chemotherapies can also have immune-dependent effects. They can evoke T-cell infiltration into tumors. They can generate new T-cell responses. So we put a chemotherapy into our murine model, 5-fluorouracil. It didn't control tumor growth as a single agent, but it did what the literature had predicted. It started to heat up that tumor microenvironment. We were seeing more evidence of CD4 and CD8 T cells infiltrating these tumors. So then we did the co-administration experiment. We used chemotherapy to heat up the microenvironment, and then we explored whether our perivascular TAMs could play a role in immune modulation of that response. And indeed, when we combined a hemoxygenase 1 inhibitor with an immune-stimulating chemotherapy, we saw tumor control. And actually, it was a really robust and very kind of clear-cut response between the single-agent modalities. I appreciate small molecules can have off-target effects, and we just wanted to mitigate this by an experiment where we did a genetic inactivation of hemoxygenase 1 within the myeloid compartment. We achieved this using a floxed allele mouse and a CD11b specific Cree. And in the Cree positive animals where we genetically inactivated hemoxygenase 1, we also found this responsiveness when we put in chemotherapy. So it was mapping what we'd seen using the pharmacological inhibition. This was also immune-dependent control, because if we depleted CD8 T cells from these animals and put in the dual therapy of hemoxygenase 1 inhibition with a chemotherapy, we found that we no longer controlled tumor growth. So indeed, the live-1 positive perivascular TAMs, through their hemoxygenase 1 expression, seemed to be able to modulate this response to chemotherapy. They were modulating this anti-tumor immune response within the tumor. And indeed, if we inhibit hemoxygenase 1 ex vivo, we could show in every single tumor that we analyzed, we could improve the effector function of the CD8 T cells within those tumors, both looking at this from a perspective of interferon gamma and TNF. Now, having to circle back now to this idea, what we actually had models in the lab whereby we could use interleukin-6 knockout mice to prevent their development, or CCR5 knockout mice to prevent their nest formation. We questioned whether, like we did with the inhibition, if we prevented a perivascular macrophage from forming, or if we prevented its ability to communicate and form nest, does this also influence the outcome from chemotherapy? And indeed, in the interleukin-6 knockout mouse given 5-fluorouracil, we controlled tumor growth. We also controlled tumor growth when we added 5-fluorouracil to the CCR5 knockout animals. And excitingly also, in parallel to what we saw with the inhibition of this enzyme, in the absence of these cells being able to polarize, the control was also lost in the absence of T cells, as it was in the CCR5 knockouts. So everything was, again, building confidence to this idea that these perivascular macrophages were immune modulatory. But also, because of this observation in the CCR5 knockout mice, it appears that also this axis is, and their kind of nest structures are also a biological attribute. So in essence, this is a collaborative subset of macrophages. We actually, on a kind of a slightly different line, but also associated with immune suppression, we stumbled across this observation that in these animals, when we immune profiled our microenvironment, this is outside of the context of chemotherapy, we actually also identified that there seemed to be, where this red arrow is pointing to, this influx of CD8 T cells into the tumor microenvironment in the absence of these cells. So this was suggesting that as well as having an immune suppressive role for T cells that were in the tumor microenvironment, they also could be playing a role in actually trying to keep these T cells out of the tumor microenvironment also. So we explored this using an in vitro assay. We set up a pseudo in vitro trans-endothelial migration assay whereby we could put live-1 positive macrophages, which were the polarized macrophages, on the basolateral side of a transwell assay. We had a barrier of endothelial cells sitting within the transwell, and onto the top of it we placed the T cells. Into the bottom well we placed the chemokine receptor CXCL10 to draw the T cells through. Now, what we found was that the CD8 T cells were also actually being restricted in their ability to transmigrate in the presence of these IL-6 live 1 positive macrophages. If we did this with a macrophage where we'd knocked out hemoxygenase 1, we did find that there was some recovery of the ability of these T cells to cross the endothelial membrane, suggesting that hemoxygenase 1 also could be providing immune exclusion attributes to these cells. We tested this in vivo also. Now, for this experiment, it's a slightly different Cree to that I've shown you previously. This is now using a live 1 Cree. We thought it would be a more specific approach for this direct subset of cells. In essence, what we are achieving here is a genetic inactivation of hemoxygenase 1 in our live 1 positive TAM subset. Reassuringly, when we knocked out this hemoxygenase 1 activity in this subset, we saw what we saw previously. If we put a chemotherapy into these animals, they control tumor growth. So as I say, everything was building this kind of confidence on this axis. But let's say, taking outside of the chemotherapy context, where we just knocked out this hemoxygenase 1 activity, you can see here that indeed we also saw a greater prevalence of CD8 T cells entering the tumor microenvironment of these Cree positive animals, which is the pink bar on the far right. My apologies, I actually haven't labeled that one. But that was a statistically significant increase, highlighting that these perivascular macrophages did play a role in immune exclusion. So in summary, what I've discussed with you today is this concept that these monocytes develop into macrophages within the tumor microenvironment. The endothelium is secreting interleukin-6, which is polarizing these cells to express live 1, CD206, hemoxygenase 1, but also connecting a communication axis involving CCR5 and its ligands CCL3 and 4, allowing these macrophages to indeed be collaborative, forming multicellular nests. And I hope I've demonstrated to you that these nests are associated with immune suppression, but also immune exclusion from the tumor microenvironment, which has implications for the response to chemotherapy. So to thank you to those that did this work, Karen Feehan, who presented with regards to some of the genetic knockout models I've presented to you today, Tamara Muliajitan, who did the work on the small molecule inhibitors, Joanne Anstey, who was pioneering these kind of nest structures and their communications, and James Opsimer, who did all of our work with pioneering the idea of these TAM subsets and identifying the live 1 positive TAM as a particular focus for us. Henrika Müller did a wonderful job setting up the perivascular nest in vitro assay. Thank you to our funders, the ERC, Cancer Research UK. They do an amazing job supporting our lab to keep our these experiments moving forward. The CRI for our work on chemotherapy and the MRC for studentship support. And thank you all for your attention.